Hello, my name is Dr. Cleo Rolle, and welcome to our series, Scientific Insights into COVID-19, what 6 months of research has revealed about this new disease. In this video, I will be discussing the scientific evidence to address the question, was the SARS-CoV-2 virus created in a laboratory by scientists? Let's begin by exploring the structure of a virus particle or virion for the SARS-CoV-2 virus. So, in the outer coating of the virus, you have envelope proteins, membrane proteins, and spike proteins. And these spike proteins are what's critical for the virus to enter into host cells. Inside the virion, you have a viral genome, which is composed of RNA and nucleocapsid proteins that protect the viral genome. And so some of the myths that you may have heard surrounding whether or not the SARS-CoV-2 virus was created in the lab were that scientists created the SARS-CoV-2 virus by combining genetic parts from multiple viruses, that scientists may have designed and genetically engineered the SARS-CoV-2 virus, or that scientists grew the SARS-CoV-2 virus in the lab from a bat virus or some other type of virus. And so our best lines of evidence to address these myths are to look at the genetic code of the SARS-CoV-2 virus, as well as the structure of some of the key proteins that are involved in the viral entry into the hosts. And so based on these, I hope that I can prove to you that we know that SARS-CoV-2 to the best of our knowledge with the scientific evidence that we have, was not created in the lab by scientists. And so if we begin first by looking at the SARS-CoV-2 genome, it's approximately 30 kilobases or 30,000 nucleotides. And within that 30,000 nucleotides, it contains the information needed to make all of the proteins that I mentioned at the beginning that are needed for the structure of the virion. So scientists begin by looking at these nucleotides, and they compared it to similar coronaviruses that they isolated from bats and pangolins, as well as other known coronaviruses that infect humans, SARS and MERS. And so what they first wanted to look at was there any evidence that scientists had taken parts of different viruses and put them together to make the SARS-CoV-2 virus. And so scientists do have the ability to take different pieces of nucleotides and join them together, but there's always evidence of that genetic engineering. There's always evidence of insertions because there are different nucleotide sequences that are unique to different viruses. And so when you cut and paste those viruses together, you will always have some evidence that there were different strains that were combined. And so when scientists looked at the sequence of SARS-CoV-2, they could not identify any evidence of genetic insertions. These were in samples that primarily they collected from patient samples. And so in patients who were displaying symptoms of COVID-19, they were infected by SARS-CoV-2, and when they looked at those SARS-CoV-2 virions, there was no evidence that they had been cut and pasted together. Then scientists also compared the sequences of these nucleotides, so the order that these nucleotides were placed in, and they looked at were the order of these nucleotides, these 30,000 approximately nucleotides that were in the human SARS-CoV-2 virus, how does that compare to the sequence in pangolins versus the sequence in similar viruses isolated from bats? And when they looked at that, they found that the backbone of SARS-CoV-2 shared about 96% identity, so the nucleotides that it contained were about 96% similar in their order with the bat virus, and only about 90% similar to the pangolin virus. And so if you think about the fact that humans and chimpanzees have about 99% of their genome share some similarity, and how different we are from chimpanzees, you can see that it's very relatively low similarities between the SARS-CoV-2 and pangolins or bats. And so it's unlikely that scientists started from these backbones and created the SARS-CoV-2 using one of those as a starting point. What's more likely is that the SARS-CoV-2 virus was generated naturally through a recombination event that happened in an animal that was simultaneously infected with the bat COVID-2 or the bat virus, coronavirus, and the pangolin coronavirus, because there is some similarity between some parts of the bat virus and then some parts of the pangolin virus. And so I've mentioned previously the spike or the S protein. And so that protein is essential for the virus to be able to enter into host cells. And it does so by interacting with ACE2 on host cells. And also another thing that plays a role is another protein, TMPRSS2, that essentially cuts the spike protein and allows the virus to then enter into the cell. And so in order for SARS-CoV-2 to be able to enter into the cell, this interaction has to occur. And so scientists decided, well, what if we were able to engineer an S protein? What would that look like in order to ensure that it was able to bind to ACE2 and enter the cells? And so they did some computational modeling, and they came up with a variety of different structures for those S proteins based on the best fit with the ACE2. And so you can think about this as a lock and key. And so they figured they had the lock already, that's the ACE2, but they needed a key that would fit into that lock. And so they designed a couple of different keys. And what was interesting, all of the keys that they designed, none of them was the same as the S protein that exists naturally on the SARS-CoV-2. And in fact, when they looked at the structure of the SARS-CoV-2, it isn't even predicted to be able to bind to ACE2. And so scientists were going to design an S protein and design a virus. You would think that they would design the best fit, because that would ensure that it would be able to enter into the host. And so we know based on computational modeling that that is not the case. And so it's unlikely that this SARS-CoV-2 was created by scientists, because if scientists were going to design a virus, they would design a virus that had a better spike protein. So scientists went on to look further at the structure of the spike protein, since it was critical for recognition and entry of the SARS-CoV-2 virus. And so they looked at the structure of the S protein, and what they found were there were two sites that were recognition and entry into host cells. There was S1, S2 site and an S2 prime site. And so cutting at both of these sites were required for entry into host cells. The S2 prime site is cleaved or cut by the protein TMPRSS2, and the S1, S2 site is cut by furin proteases. And so in order for the virus to be able to enter the cell, the S protein has to be cut at both of those places, primarily the S1, S2 site. And so when they started to look, they found that this S1, S2 site was not something that was unique to SARS-CoV-2. It was already previously reported in MERS. And so this was not some novel feature that scientists had created. It was something that was already naturally occurring in another virus. And so that is very strong evidence that there's no way that scientists could have known that putting this cut site into an S protein that's not supposed to recognize a human ACE2 receptor would be such an effective virus. The findings to this point, scientists did not detect any evidence of genetic engineering in the SARS-CoV-2 virus. There was no evidence that a scientist in a lab somewhere had cut and pasted different strains of viruses together to make some super virus. Also, when they looked at the receptor binding part of the spike protein that is critical for recognition and entry into host cells, scientists did not come up with the structure that SARS-CoV-2 actually uses. They came up with different structures that have even better binding ability. And so if scientists were going to spend time designing a virus, they would have predictably used a virus that should used a S protein that should bind best. Also, when they looked at the structure and the cleavage sites that are critical for entry into viruses, they found that the furin protease cleavage site was not something that was new and newly created in SARS-CoV-2. It already occurred in nature. And so it was more likely that there was some recombination event happening to allow the generation of the SARS-CoV-2 virus. And so that's just some of the data that refutes the notion that the SARS-CoV-2 virus was designed by scientists in a lab.